for you? Okay, so let's get started. So welcome everybody. This is one more session of this Euroglia webinar series where we all join together in our love for microglia. So before we get started with today's session, I'm gonna leave the room to Philip de Jager. I'm gonna share a slide for him. Here it is. So Philip, it's all yours.
Great, thank you, Amanda. So, hello, everybody. So, Amanda kindly sort of allowed me to spend a few minutes here introducing a new project that we're launching, thanks to support from the Chan Zuckerberg Initiative. What we want to address is the basic idea is to build a human microglial resource for the community. The challenge we're trying to address is of course, the question where many groups have now generated data of different types relating to human primary microglia and different induced microglia. And each of us has done different types of clustering or other methods to try to understand the heterogeneity of these cells. But of course, everybody does it a little bit differently, and it's a little hard to understand exactly what each group is seeing and how these different clusters or different signatures relate to one another. So what we propose to do and got a moderate amount of support to initiate is an effort to bring the community together to try to have an initial effort to create a resource where we would have one data set that could serve as a reference, and then we can develop a cluster-based method or another type of factor-based method potentially to try to establish a reference framework. And then the community could use this to try to integrate and compare results amongst each other. Because the idea is if we all project into the same reference, then we can actually see exactly what's happening relative to the different studies and also across diseases, since often people only look at one disease, for example. So next week on May 13th and 14th, we're going to have two introductory sessions. They're the same, so you only have to attend one of them. One is in the morning Eastern time in the US, that's on May 14th. And on the 13th, we have one later in the day at 5 o'clock. This is more for the West Coast people. And yes, and so again, this is where we'll have the introductory session. We're also going to tag a website with the information to the Twitter handle for this meeting. So in case you want to look for the information there as well. And this QR code, if you scan it, will bring you to a website where we've outlined the project. And again, the next week we'll have the initial meetings to explain what we're trying to do and to see who is interested in participating. This is really meant to be a group activity. So I think I'll just pause there. And again, through the website, you can actually register, which is just simply to let us know who's interested so we can then establish a mailing list.
Thank you very much, Philip. I'm gonna stop sharing. I have to say, we all think it's a terrific initiative, and I hope as many people can join you as possible. And we're also expecting to hear a little bit more about this initiative at the EMBO Microglia meeting that most of us are attending at the end of this month in Genoa. So without further ado, now I'm going to introduce the team that organizes all these series. You know most of us. There's a team from Lausanne, led by Rosa Paulicelli, and we have Anne-Claire Compagnon, Fanny Martineau, who she will be handling the introduction today, and Giorgio Corsi. And from Achucarro, we have myself, Fede Soria, and Marta Pereira, who will be handling the questions and answers. And you can see we really have a terrific lineup of speakers. So without further ado, I'm gonna leave the room to Fanny. So welcome, Fanny.
Thanks, Amanda. So our speaker today is Dr. Miao Jing. Miao received his PhD in 2018 from Peking University, and he joined the Chinese Institute for Brain Research as an independent research fellow already in 2019, and was promoted very rapidly as principal investigator in May 2022 after international review. His lab's research combines technique development and applications to decipher the dynamics of extracellular signaling molecules in information encoding, processing, and transmission, especially in the context of brain-neural immune interactions. He's now serving as the leader of projects in China, as well as the Intergovernmental Collaborative Project. Miao, the floor is yours.
Hi. Yeah, thanks so much for this kind introduction. And let me share my slide first. Can you see that full screen slide?
Yes.
Yeah, so really thank you for Amanda Rosa for organizing this great symposium webinar and inviting me. Actually, I'm a newbie in this field, so really hope to share our thoughts on recent progress as well as promoting some new collaborations, potential collaborations in the future. So what I would hoping to tell in the next several tens of minutes is kind of interaction between glial cells in the injury response. What we call homeostatic injury response. I hope in the end of the talk, I will be able to convince you that this kind of injury response still need to maintain its own balance by its internal regulation. So before I go into the detailed project, I would like to briefly summarize our lab's interest. So we are very interested in deciphering the so-called syntax of cellular language. We all know that the brains or many organs contains multiple cell types, and they actively communicate with each other, just like the human communities that through different molecules. So the one limitation or one difficulty for this cellular language is that they need to encode unlimited information with limited codes, with limited variables. So just like deciphering the Morse code that originally used in the human society, so this can be achieved by using different properties of these molecules. Like we can use different variants like dots and bars, as also changes in their latency, arrangement, frequency, as well as the combination of different codes. So indeed, the biological system also can use not only the absolute level of one molecule, but also the different properties of that. For example, the duration of the signal or the rising slope or the frequency of that signal. So what we hope to understand in the lab is to have a detailed look or detailed analysis of each molecule involved in cellular interaction, crosstalk, and to see how their properties enable them to encode specific information and how that information get passed to the receiver cells for selective function. So today I will actually talking about an old topic, which is the brain injury. So we all know that brain injury is kind of prevailing pathological situation that can be initiated or accompanied with various diseases, like traumatic damage, virus infection, or neurodegeneration. So the invention of new therapies or treatment for these diseases often relies on the first understanding of the basic mechanism that lie in how the brain are able to encode and sense this injury information. However, there is not fully understand because most of the time, these injuries are classified into the so-called pathological situations or simplified as disease. So they are often put in the opposite side with the so-called health, which is defined as the absence of disease. Therefore, the ways to study the health or the physiological situations emphasize the importance of homeostatic regulation, while that usually be ignored in the study of pathological situation or especially injury. So that leads to some of the current view simplifies the brain injury response as a kind of passive consequence of this damage, just like the leaking of the water from this broken glass bottle. So in this way, we actually, most of the treatment are trying to target the input or the injury itself. Well, that kind of ignore that the brain can maybe is able to sense and translate the injury information into its own language and use that language to pass or transmit the information to guide the downstream cellular response. So indeed, recent several elegant papers as well as reviews already pointed out that this kind of injury response in the brain is highly organized in time and space. So with different molecules or cellular events initiated and terminated at a selected time and space. Also, this kind of injury response usually matched with the initial injury properties, for example, its intensity. So all of this suggests that the brain are not simply waiting there to be damaged, but actually can actively sense the injury and use its own ways to translate that. So we are curious how the brain are able to, at the beginning, to sense the injury and encode that with its own language. So to study this, we try to find whether any of the extracellular molecules might be important as an injury kind of representation. So previously already research already shown that there's a lot of molecules that can be accumulated after injury. Most of them are passively leak from cells, such as ATP or cytokine chemokines, as well as some DNA fragments or lipids. So among them, we are very curious or focused on this cholinergic molecule, ATP, that has also been frequently talked in this previous seminars. So elegant works have shown that using bioluminescence assays that upon this spinal cord injury, there will be increase of bulk ATP signal concentrated around this injury region, suggesting that this ATP signal is indeed associated with the injury. Well, also this seminar.
Miao, can I stop you for a second?
Yeah.
Sorry. So some people is hearing a weird noise on the microphone. So maybe there's some movement, or maybe you can grab your microphone a little bit closer. Do you have a microphone?
How's that?
Now it's okay. But sometimes when you speak, we hear like a beep beep.
How's that now?
Yeah. Now it's perfect.
Yeah, how's that now?
Yes.
Okay, okay. Maybe I just stopped moving my head.
Yeah, I think it's related to the movement. Okay, thank you very much.
Yeah, okay. Yeah. I will stay still. Sorry for that.
I think it's related to
so
the
I will
cable.
Continue. Yeah, okay, thank you.
Sorry about that.
Okay, so I will continue with the introduction. So I'll just mention that this ATP has been shown to be associated with the brain injury, and that also correlates regulate microglia behaviors. For example, the previous Professor Garen-Biao's group has shown that the rapid laser injury in the brain can recruit the microglia process that to extend rapidly to this injury center. And by applying this ATP degraded enzyme APRIS, they show that this microglia migration are largely blocked. So all of this supporting the importance of ATP as a kind of damage-related molecule. But without looking detail into this ATP dynamics, we do not know whether they are actively released or kind of passive leak from the intracellular high concentration of ATP, and how they be able to encode information of the injury. So indeed, to solve this is really critical to real-time image the ATP inside the brain in the native situations. So the technique is limitation is how can we detect the ATP in time and space. So really fortunately, during my PhD period, I have been working together with Professor Yulong Li at Peking University, where we developed the so-called GRAB sensors, short as GPCR activation-based sensors, that can enable the real-time imaging of specific neurotransmitters or glial transmitters into convert that into a fluorescent output. So just a short introduction for these sensors. We use the endogenous G protein-coupled receptors as the backbone. For example, to sense ATP, we will use the P2 receptors, and we will fuse or engineer a fluorescent protein, specific fluorescent protein, into the loop of the GPCR, and then to sense the conformational change of the GPCR when it binds to the ligand. Therefore, real-time imaging could be used to capture the fluorescent changes and then reflect the dynamics of selected neurotransmitters. So previously, we have engineered such sensor for acetylcholine dopamine, as well as some neuropeptide recently. So also recently, in collaborations with my previous advisors, we engineered and validated these new ATP sensors called GRAB ATP 1.0, which I will use throughout this talk. And as you can see, they express well in the culture neurons and can show a dramatic increase of fluorescent signal upon the application of extracellular ATP. So with these tools, we'll be able to express that in the mouse by AAV dependent viral expression. And we also use a kind of focal laser ablation model shot as FLA here to selectively ablate the local regions in the center of the image region. Therefore, we can image the ATP dynamics before and after the injury and at different positions. So next, I will show you the real video of the ATP dynamics inside the brain. So before this laser injury, there's quite constant ATP levels. But after this FRA injury, you can immediately see that there's a lot of spatial temporal selective ATP events that has been first observed during due to this technical improvement. So by analyzing these signals, we found there are at least two types of ATP dynamics or ATP signals. One we named as ATP burst, which is concentrated on the injury and quickly disappeared within 20 minutes. And it's show a limited propagation outside. And another signal is the flashing type of ATP events that you can see over the imaging regions and occur at a different time and position. So we give it a name with the individual flash event as the so-called inflare, short as injury-induced flashing ATP release. So by analyzing this fluorescent signal changes of the sensors, we could capture the individual signals at different time and extract it out as for further analysis. So indeed, we could find that after this one shot of laser injury, there will be one immediately burst happening, very strong ATP release locally, but then quickly disappeared within around 10 to 20 minutes. While there are multiple iterative generated inflares at selective time, and they occur at a flashing pattern where we can analyze the progression of this signal over time and space. So we can also quantify the properties of individual signals. For example, the individual burst, the burst signal of ATP are significantly larger than individual inflares, and it also lasting longer in duration over individual inflares. While the distribution of individual inflares show a single peak, potentially point out they are likely to be homogeneous in properties. So without showing too much control experiment showing that this signal indeed reflect ATP dynamics, I will quickly go to the first question, is that we're curious whether any of these signals fits the criteria as the internal injury representation. So all this question can be rephrased as what is the criteria for so-called injury representation? So actually in this field, there has been a lot of observations that showing the migration of microglia as an index for this internal injury response. So we also use the migration of microglia as a characterized of this injury response internally. So we found that actually there is a large, huge spatial temporal gap between this injury itself and the microglia response. Specifically, when we give a transient laser injury that only lasts for 2 seconds, that can be able to mobilize the microglia over 12 or even 24 hours. That is kind of persistent in time. This microglia labeling is through this CX3CR1 JP mice, transgenic mice. So that difference in time indicating that a temporal gap between this transient injury and this persistent microglia response. Also, we'll be able to, when we analyze that in space, we'll also find that this local laser injury that is only 50 microns are able to mobilize microglia over a large region globally, and even microglia that are 300 micrometer or several hundred micrometer away from this injury center are also be able to sense this injury and change their process directions. So all this suggests that not local microglia process may sense the local ATP burst, but the microglia surrounding far away from this injury need to have a kind of internal signal that we name as injury representation that to bridge this kind of spatial temporal gap. So to next question, to next wonder whether any of these ATP signal can be persistent and widespread, we conducted we first conducted the long-term recording of these inflares at different time point after laser injury. So indeed, we found that consistent with the data I previously showed, that this ATP burst is quickly disappeared and never regenerated again. However, this inflares could be observed even after 12 hours or after 24 hours after laser injury. So there could also still see these flashing events surrounding these injury regions. So supporting that these inflares are much more persistent than this local ATP burst signal. Also, at a spatial distribution, we'll be able to use the enlarged field of view during two-photon imaging and see that these ATP bursts are concentrated around this laser injury site, while these inflares can be widespread in space, that it will be able to broadcast this injury signal over the space. So altogether, we concluded that this inflares, with its persistence in time and widespread in space, that kind of fit the criteria for bridging this injury to this microglia response. So when we next immediately ask the second question, how inflares, if acting as the injury representation, be able to encode the injury information by its own properties? So that's also, as I just mentioned, is also the interest of our lab research. So in principle, these inflare signals can encode the injury properties by different methods or different strategies. One strategy we named as the so-called amplitude coding is by changing the level or size of individual inflares. So this is kind of encoding the signal at individual single event level. Or just as the action potentials in neurons, these inflares can encode the injury properties at the population level with changing its number or changing its frequency, while remaining the individual signal unaltered or digitized. So to further dig out the real encoding strategy of these inflares, we perform a detailed analysis and plotting these inflares as this bar graph, with each bar representing a single inflare with its height representing the indicated the changes, the fluorescent change of the signal or the amplitude level of ATP release, and the duration means the length. The length means the duration of the signal. So we next apply the different intensities of injury to this to record the inflares in vivo. As you can immediately see that when we give either weak, medium, or intensive injuries, each inflares seems to be relatively maintaining in the same diameters, while their frequency or the number progressively increase over the changes of injury intensities. So that can also be supported when we analyze this signal at a single event level using this bar graph plot, which each line representing data from single mice, or analyze the frequency distribution that representing the changes of frequency upon different laser injury intensity. So all of this suggests that inflares are digitized, or each individual inflares may act as the fundamental signal unit while they organize in encoding the injury intensity at the population level by its frequency change. So to further support this, we also analyze the properties of single inflares at different injury intensities, and we found that indeed single inflare properties are relatively the same, and putting all them together cannot be able to segregate or distinguish the injury intensity we applied. Well, if we use the population frequencies, we can see that clearly distinguish of inflare frequencies among different injury intensity groups, and we can now separate the different intensities based on the frequency properties of these inflares. So to further support this, we actually conducted built a linear regression model trying to retrospectively predict the injury intensity from the properties of inflares. In this linear regression model, we analyze the individual properties as well as the population properties of inflares, and we give them different weights and trying to find the best model that can predict the laser injury we applied. So a successfully trained model predicted well of the laser injury intensity from the inflare properties, and the top weighting parameters are all belongs to the population level of population properties of inflares, especially the frequency. So indeed, if we exclude the frequency from this model, or we use the shuffle data, that is to shuffle the pairing between frequency and the laser injury intensity, we all get a poor prediction of this injury from these inflares. So therefore, this kind of quantitative relationship between these inflares and injury ensures that inflares can accurately or robustly encode the injury intensity. While this linear relationship, I will go back later when I talk about these homeostatic ideas. So just now I mentioned that these inflares are able to encode injury intensities, but we're also curious whether they are also be able to encode other properties of injury. For example, this position of the injury that is very critical for this microglia to conduct directional migration. So as you might remember from this video, it seems that these inflares are relatively random in space. However, when we project the inflares over several minutes, we can see that they actually form a so-called density gradient, which also means that close to the injury, there is a higher probability to initiate an inflare. So such kind of population or density gradient can span over hundreds of micrometers and also can last for 12 to 24 hours. That means they can continuously provide the direction to guide the microglia for the sustained migration. So we can further show this by tuning or switching these injuries into different positions. As you can see, switching these injuries to position 2 or 3, that resulted in the falling of the inflare gradient to this injury center. While we can also extract the gradient center from these inflares and align that with the injury center we applied. And we found that they are close with each other, further supporting that extracting the information from this inflare gradient can predict or can decode the injury position. So of the previous work, I have been showing that these inflares might be able to encode information by its own properties. So next, we wonder how are inflares generated at the molecular and the cellular level. So as ATP can be or has been shown to be released from either astrocytes, microglia, as well as from neurons, we first conducted a cell type specific screening by ablation or silencing different cell types locally in the brain and trying to record the laser injury induced inflares at this background. So successful ablation of local astrocytes through either genetic methods or HSV-TK by using HSV-TK system or using this gliotoxin L-alpha-A are successfully ablate local astrocytes and resulted in no increase of inflares after FLA. So supporting that astrocyte might be the major contributor for releasing inflares. Indeed, similar experiments were conducted to ablate local neurons by expressing this caspase in neurons. As you can see, ablating local neurons did not affect the laser injury-induced inflare release. And also using the CSF1R inhibitors to deplete microglia also did not affect the laser injury-induced inflare increase. So within these astrocytes, the intracellular calcium serves as the major signaling molecules, at least the field agrees. So we next wonder whether this ATP release is kind of calcium dependent. So using this imaging method, we first check whether there is increase of calcium signal. Indeed, this is conducted in brain slice ex vivo. So when we give a laser injury, we see there is a progression, there is a propagation of calcium wave initiated from the laser injury center and rapidly propagated to these surrounding regions. So that indicating the correlation or coexistence between this calcium wave and inflares. So to further prove this necessary of these calcium signals, we use subsigagin to deplete the intracellular store, or use this KLEX developed by biologic groups to deplete the calcium, to decrease the calcium level in the astrocytes specifically. And we can all see that decreasing or silencing this calcium signal in astrocyte resulted in no increase of inflares after FLA. So conversely, if we use chemogenetic method to elevate calcium in the astrocyte, and in the absence of injury, we'll also be able to increase the inflares even without injury. So that supporting that this calcium signal is really important for the generation of astrocytic inflares. So finally, we would like to answer what is the molecular mechanism of astrocytic ATP release. So we searched for the literature and identified several molecules that has been reported for mediating astrocytic ATP release, including several membrane channels like pannexin-1 volume-regulated ion channels, as well as the VNAT that is important for transporting this nucleotide into these vesicles and for this vesicular release. So we first conducted the pharmacology screening by applying different blockers for these selected molecules. And we found that several molecules that are able to block this pannexin heavy channel, including the probenecid acid, short as PPN, as well as this peptide inhibitor or another antibiotics, all are able to block ATP release after FLA. While this control scrambled pannexin peptide do not have the blocking effect, as well as other blockers for the several channels or VNAT. So this pharmacology experiment result was further affirmed by the genetic methods, where we generated the pannexin specific cell type specific pannexin knockout in astrocytes, which greatly reduced the FLA-induced inflare increase. While in neurons, if we knock out pannexin in neurons, they do not affect this inflare generation. Conversely, if we overexpress this pannexin in astrocyte, can boost this inflare release after same FLA injury. So overall, we concluded that this type of spatial temporal selective ATP release, which we name as inflares, are kind of selectively released from astrocyte, at least in our context, and that is in a calcium-dependent manner through this opening of pannexin-1 heavy channel on the membrane. So with this molecular mechanism, we'll be able to address one fundamental question here, that is whether the information encoded by inflares are really critical for microglia. In other words, whether microglia decode information from inflares or they simply decode that from the initial injury. So we designed one experiment where we give the same laser injury, but we tune the inflares by either knocking out pannexin-1 that resulted in a decrease of inflares, or with overexpression of pannexin-1 that resulted in elevation of inflares. As you can immediately see from this microglia staining, the same laser injury resulted in different microglia accumulation at this laser injury site that follows with the pannexin-1 or with the inflares level after with the individual manipulations. So we can also see their relationship between these inflares and the microglia number at the center, where we see that they follow a constant linear relationship, further supporting that microglia are able to directly retrieve or decode the signal from these inflares. While if we change the inflare encoded signal with this injury by overexpressing or knocking out pannexin-1, there is a loss of connection or loss of matching between this microglia with the initial injury. So this supports our view that this microglia are major information of microglia are decoded from this inflare encoded signals. So what about this injury outcome? So we use this local cell apoptosis as the index to roughly estimate the local injury outcome. So similarly, if we give the same laser injury, but tuning the inflare properties or inflare encoded signals by knocking out or expressing pannexin, we see that the local cell apoptosis follows the inflare encoded signal rather than following the initial injury. That this resulted in a similar matching between these inflares, between this cell apoptosis with inflares rather than with the initial injury. This also supports the critical view that the abnormal matching between this injury to inflares or inflares to microglia will result in changes in function in the injury outcome that might be the real cause of disease, which I will talk about later. So we also analyzed several properties of this microglia, including their morphology, showing that analyzing the branching point, branch length, as well as the cell body volume. So this figure might look a little bit complicated, but you can see that the application of laser injury changes the overall microglia morphology that could be partially reversed when we block the influence by using Pinaxin inhibitors or conditional knockout of Pinaxin-1 in astrocytes. So we also checked other properties, functional functions of microglia, like phagocytosis, that also be influence dependent, as well as the proliferation that is also depends highly on the generation of influence. So there's one leading question on this cell apoptosis, that is whether this influence directly regulate the neural apoptosis through like P2X receptors, or indirectly through this microglia dependent signals or microglia changes. So we conducted an experiment in the background of microglia depletion through these CSF1 inhibitors. And indeed, we found if microglia are depleted, this secondary injury demonstrated by the cell apoptosis are no longer high as the normal situation, as well as the Pinaxin treatment or by blocking with PPN lost its effect in changing this cell apoptosis numbers. So that indicating this so-called secondary injury indicated by cell apoptosis is largely microglia dependent. So I would like to have a brief summary of the past things I just mentioned. So we found that there's actually an internal mechanism to actively sense and encode the injury signals through this spatial temporal selective ATP release coming from astrocytes. And also this astrocyte release the influence, the ATP signals match with the injury properties that have been encoded injury properties, and they can transmit the information to microglia. And this kind of mismatch in this information flow might lead to changes in injury outcome. So with this, we actually coming with an immediate question, that is how they fit so well with each other, or how influence are able to match with the injury properties so well. So if we can imagine that if astrocyte itself or having this own function, they may easily get overactivated, that lead to exacerbated injury damage, injury outcome. So we reason that there might be some other mechanism that trying to maintain this homeostatic balance for this ATP release or for this injury signal transmission. So actually, from our previous result, there's already some hint that microglia, the main topic for this webinar, and I hope most of the people here are already acknowledge that its microglia are very active in their cellular decision making, as well as the intracellular signals. So besides working as a simpler receiver for these ATP signals, microglia might actually participate in this regulation of injury response. So one fact that lead us to think of this is because the accumulation of microglia after laser injury is highly correlated with the termination of influence. That actually makes sense because the influence, the properties of this, the function of this influence are to recruit microglia. And when this microglia are already there, they probably need to stop this generation of influence. So to further support the microglia are indeed able to participate in the regulation of ATP release, we conducted the manipulation or ablation of microglia through these CSF1 inhibitors, and also we let them regenerate by the remove of this drug. Therefore, we can record this laser injury-induced influence ATP release in different microglia backgrounds. So as you can see quickly from this video, compared with the control conditions that with microglia, the microglia depletion resulted in a lot of significant elevation of influence numbers. While if we let the microglia to be regenerated, there will be the influence or the laser injury-induced influence release will get back to the normal situations. So that's suggesting that indeed the microglia might be able to give or provide a negative feedback to this associated ATP release. So to further confirm this, we try to use different methods to alter the selective states of microglia. So we first use this P2Y12 knockout. With this P2Y12 is selective expressed on microglia as important for sensing ATP and initiating the downstream like microglia cellular response as well as its migration. So compare with the wild-type mice that with P2Y12 on microglia, P2Y12 knockout mice show elevation of laser injury-induced inflares. That is suggesting this microglia feedback might require its first activation by these ATP signals. As another evidence, we try to use this chemogenetic methods in collaboration with Adam Dennis' lab in Hungary. So we try to express selectively the chemogenetic tools in microglia. As you can see, compared with the CNO alone control group, application of CNO in this HM3 expression microglia mice resulted in a decrease of FLA-induced inflares. So that means this chemogenetic modulated microglia increase their ability to feedback to this astrocytic ATP release. So combining this increase and decrease method, we want to emphasize that actually this microglia can achieve a state-dependent feedback that they themselves are able to tuning their feedback level based on their cellular states or properties. So whether that contribute to this homeostatic injury response, that is to maintain the ATP release at a selective balance. So as I previously introduced, these inflares can able to encode different laser injury intensities that we can be able to conduct similar experiments by giving different laser injury with at the background with or without microglia. So from this data set, we'll be able to calculate a so-called microglia-dependent inhibition. That is the difference between these two groups among at the individual FRA intensity applied. So we can imagine that if for a homeostatic system, an increase of input, which means more inflares, should require a stronger inhibition from this potential microglia. So that is indeed what we can observe from this inflares correlation with relationship with this microglia-dependent inhibition, that they follows with a positive linear relationship. Therefore, this kind of sophisticated microglia cells are able to tuning their ability and change their feedback level depends on the signal they receive from this astrocyte. While on the other hand, astrocyte, when receiving this negative feedback from microglia, can adjust their releasing of ATP. So these two cells actually form a unit or a functional unit where they coordinatedly to maintain this so-called homeostatic injury response in the brain, with each cell not only as the signal outputter as well as the receiver. So I will briefly mention our effort to analyze the downstream or the underlying molecular mechanism, which we are still working on to make it to understand this full picture. So we found that we hypothesized that the microglia might need to release something to working on this astrocyte, since they are not physically close with each other in most of the situation. So to find these potential molecules, we first used the brain slide system and we applied different molecules, especially the cytokines and the chemokines from microglia, and to see whether they affect this FRA-induced laser injury-induced ATP release. As you can see from this several screenings I show here, this application of IL-1 beta, which is kind of inflammatory in the field, but can actually silence the FRA-induced ATP release. And this effect could be blocked by this IL-1 receptor antagonist or by IL-1 beta neutralized antibodies, which I did not show here. So to further confirm this IL-1 beta is indeed released from microglia, we conducted the knocking out of IL-1 beta from microglia, or knocking out of IL-1 beta receptors from astrocytes through transgenic mice. As you can see here, this red lines indicating this knockout mice, and compare with the control mice, this laser injury-induced influence are significantly elevated, not only in the absolute amount, but also in this duration or this persistence time after laser injury. So this altogether support that the absence of IL-1 beta receptor signaling will make abnormal or unpairing laser injury response. Or in another word, this internal system really requires the crosstalk between microglia and astrocytes to maintain this homeostatic injury response. So finally, we also curious whether these have any relationship with disease models. So we use this ischemic stroke model in mice, which is the MCO model, to see whether in this situation we can observe changes in influence and whether targeting them could be disease relevant or disease beneficial. So as you can see in this MCO model mice in the cortex, after 12 hours, there is excessive influence happening a lot or significant over frequency significantly higher than the laser injury models, suggesting there might be already abnormal injury transmissions in these situations. So ideally, if we can block this excessive influence, that might be beneficial for this disease. So if we can use these pannexin inhibitors, or I did not show here, we can also use this pannexin knockout mice, which we generated in knocking out pannexin astrocyte, that all shows a beneficial effect for this reducing this tissue injury after stroke in mice, indicated by the decrease in cortical infection, as well as the better recovery of these mice behaviors, showing in the open field test or the balance test. So that shows that this indeed abnormal signal transmission mediated by influence could be the real reason for the progression of disease, and as well as targeting them might be beneficial for treatment in the future. So I would like to summarize what I've told you today. So we're trying to analyze the signal transmission between different cells, and we indeed identify one type of spatial temporal selective flashing pattern of ATP release, which we name as influence, are critical for encoding this injury information and pass that to microglia. And at the same time, microglia not only act as a receiver, but they also actively participate in the regulation of ATP release. So they altogether generated this homeostatic injury response, and targeting them, or either pannexin channels or the IL-1 beta, which I mentioned, might be important for the future disease treatment. So we actually also see that influence in multiple diseases like LPS-induced systematic inflammations or in hypoglycemia. So we of course wonder what are the downstream or what are the upstream signals that cause this astrocytic ATP release, or whether we can target them in different diseases to try to have some beneficial effect. So with that, I would like to end my talk with acknowledgement of this my lab members, especially Chen Yue and Peng Weiyuan, for conducting this ATP release, as well as the microglia feedback project, together with other lab members. So also thank our collaborators, Adam, Guoqiang, as well as Yulong, for a lot of valuable inputs, and thank our funding agencies. So thank you. I would like to take questions.
Thank you very much, Ming. This really was impressive. I think you really created a new way of discussing and understanding microglia and astrocytes dialogue in health and in disease. So
thank you.
We probably have some questions from the audience. I do have some, but I would like to ask Marta to deal with them, and you let me for the end, Marta.
Mm-hmm. Perfect. So, well, remember everyone that if you have a question, you can either write your name in the chat and we will unmute you, or you can write your question and I will read it for you. So, yes, we have the first one from Lia Calcines, and she asked, do you see any sex differences in these injury responses in either the microglia or the astrocytes, or both?
Yeah, I would say that is definitely a great question. I know the field, especially the microglia field, are very curious for this sexual dimorphism. And indeed, although we did not systematically characterize the difference between sex, we found that the sex do play roles in the releasing of influence from astrocytes. I do not have the direct answer for the downstream for the detailed mechanism, but we do have the observation, and we are eager to explore that in the future.
Then we have the next one from Zeynep, and he's wondering, can we measure that calcium levels with patch clamp?
I guess you mean that whether we can measure the because this calcium in astrocytes are mainly coming from the intracellular store, so they are maybe not current passing through this membrane. So we typically use the calcium imaging to show these changes of calcium intracellularly. But for this ATP release, actually they are through these membrane channels. So there already been elegant works trying to record this pydexine channel current to show whether they are activated, whether they are able to release or able to open at different forms. So indeed, the ATP actually can, the changes of opening of pydexine channel can actually be monitored by electrophysiology method. That is also one important aspect to going through detail into this biophysics of this channel to see why they open at a certain state, a certain status that generated this digitized influence.
Mm-hmm.
Yeah, I hope I answered this question.
Then Trevor Owens asked, do microglia show developmental age-related differences in their modulating ability? And he's also wondering the same about astrocytes.
I hope I previously answered this sex difference. So these are all great questions. Unfortunately, we really have limited data directly pointing out whether developmental changes as well as the age contributing to this injury response. But if I can guess, I would say that the age definitely contribute, because that will affect the general already been shown to affect this internal injury response or be able to modulate microglia. So I think we can get old mice for a quick test, but unfortunately we do not have the data yet. From the astrocyte side, is also people already showing that this astrocyte have this age-dependent phenotype changes. And one related question with the development or with the region specificity, like either whether different brain regions have the different similar response. I think they are all what we can do further. But we hope to get these pipelines to work on this interaction between microglia and astrocyte that can be further tested, like in different sex, in different development stage, in different age, or in different brain regions. Either we find the difference or we find the similarities that can push our understanding for this internal injury response.
Yeah, sure.
Thank you.
Sorry for some hand-waving question, because we really don't have this direct evidence, but that's my guess.
Thanks.
Maybe Amanda, you can go ahead.
Okay. I do have a couple of questions, a lot actually, but try to be brief because it's almost 6 o'clock. So, and it's really, really late for you, almost midnight. And so
that's okay. That's okay.
yeah. So
don't mind that.
These inflares are a little bit like the units of this language, of this syntax, right?
Mm-hmm.
At the cellular level, why do they have this particular size between 15 and 20 microns? Does it correspond to a single astrocyte? Or what do you think?
Yeah, okay. I think that will be whether we can understand the mystery of the God's design. So I would say that for direct answer, for direct answer, so the influence size, the maximal size is roughly around a single astrocyte.
Right,
but that is the after the propagation of this influence in the extracellular space. So if we can map this original releasing site, they actually map mostly to this astrocyte process. So we think that most of these astrocyte process are generating this influence. Well,
So
that
it's
is
not like
related
coming
to,
from this
yeah.
from the cell body, like integrating different signals. So it's maybe one of the distal processes. Really, that's
Yeah,
interesting.
at least I think we still need to have direct evidence, but from our imaging data, if we extract the signal center that matches more with this astrocyte process.
Can you do
whether
like yeah, sorry.
Mhm,
Can you do like correlative imaging, like do the 2-photon and then do even electron microscopy? Is that possible?
Yeah, I think that could give us some information. So what we are doing now is to use fluorescent proteins to label the entire astrocyte morphology and to see where is the influx generated. So the difficulty for aligning this live imaging with this electron microscope will be how we can register the two images. I think that for now, I would prefer that the process is relatively generating more influx. But I think that you also pointed out one important question is whether the astrocyte can integrate some information, likely from injury, also from internal state, also from neurons, to generate influx. So that is one direction we are actually working on, is how the astrocyte upstream of astrocyte is like from the injury, how that translate into the calcium signal.
If they are facing two injuries, right? What's going to happen? Because sometimes, like, imagine you actually have I don't know, a car accident. It's not going to be just one single damage. You're going to possibly have hits in several places, and somehow this needs to be integrated. No?
Yes, that's definitely true. So I think there is a kind of integration in this single astrocyte as well as the population, because there is a kind of calcium wave that is, I did not show the data here, that is dependent on gap junction. So this astrocyte actually form a functional unit to sense the kind of injury. But in different situations, like in stroke or in this systemic inflammation, I think the signal sense by astrocyte might be different. So there's a lot of computation coming from the astrocyte side, as well as coming in this microglia side.
And then I have another question. So I really love how you put conceptually this syntax in terms of frequency and amplitude and so on and so forth. But I was wondering if there could be also a spatial language related to the space, not only to locate the site of the injury, but also maybe there could be some sort of a spatial correlation that allows not only to locate, like I said, the center of the burst, like you said, but also codification in terms of space. Do you think that's possible, or
I think you
like some sort of
what you mean is
spatial encoding? I don't know.
Yeah, I think we show some results that the influx at population level can form a density gradient. That means there are more flashes closer to the injury and less flashes outside of the injury. So that kind of gradient is a probability gradient rather than the previously thought is a concentration gradient generated by this astrocyte ATP. So this kind of probability gradient actually can encode this spatial information. As we show, if we switch this influx gradient to another site, if we switch the injury to a different site, this gradient perfectly follows with this laser injury. So I do not show here, but we also.
But in terms of the distribution, like it may not be as random as it seems. Like you have all these influx incoming, and there may be something else related to in which order or how far from each other each influx is happening.
Yeah. Okay. So
I guess this is more difficult to extract, but
I agree with you. That is also that's also important. So what we are analyzing, let me put it this way. So what we are analyzing is at the population influx level. So whether individual influx they are correlated or they are connected with each other. Like one influx will generate more influx surrounding locally. That is a kind of network things. So we didn't analyze that yet, but I think that is a good direction to go. That is whether we did observe that this influx can generate a local additional calcium increase because there's ATP receptors locally. So I think that will one influx generation will kind of promote the local generation of secondary influx. That end with the contribution to this probability gradient. But we do not, we still haven't analyzed the connection between individual influence. What we are showing at a spatial scale there is their overall together can form this gradient.
Okay.
Yeah, I agree with you.
No,
That is great,
you're right.
great point.
I think,
Yeah.
Marta, we have a related question from the audience.
Yeah.
Yes. So the last question is, do you always induce the
Mm.
injury into the same anatomic areas? I'm not doubting the glial role, but just wondering if neuronal circuits could be part of the signaling.
Yeah, okay. So I thought that is the microglia webinar, so I could leave the neuron apart. But so it's great to hear this question. So all of my talk is actually focusing on this glial cell crosstalk. But indeed, we do not leave these neurons apart. It's actually possible that this local neural input or local neural architectures or interaction between these neurons and the glial cell could be contributing to this network. So I also would like to share one of our ongoing information work is that we found that overactivating of this neuron could, in the network level, could also promote the astrocytic release of ATP. So we think that is indeed important upstreamly, that this information could coming from like either injury or coming from abnormal neural network changes. Or the downstream, they can modulate microglia, as I have shown here, but also could be able to modulate the local neural network. As a lot of paper has been showing ATP as well as adenosine can be a feedback to the presynaptic terminals. So I agree with that these anatomical areas like different brain regions or different neural circuit will be important. But I would say that the internal injury response is so important that there might be a conserved or universal mechanism. So I think the principle or the major architectures will be relatively the same, but will be further tuned by different regions.
We have one last question from the audience. We have to close down the session, but Marta, go ahead.
Yes. So are astrocytes still coupled after injury? Does gap junction coupling affect inflare activity?
Yeah, okay, thanks for this question. So actually, I did not show, but this gap junction is really important for initiating the calcium wave propagation. And without this calcium wave propagation, the outside regions cannot initiate the inflares. So the short answer is gap junction is critical for this inflare generation. But after this calcium wave, the inflares can still be generated for a long time. That does not require the further generation of calcium wave or further gap junction requirement. So I think this indeed gap junction is critical. So as I just said, these astrocytes are actually formed in a functional domain connected by gap junction.
Okay, thank you very much, everybody. I think it's time to close the session. Thank you, Miao. It was really an amazing
That's
talk.
really great.
People,
thank
you can turn
on
your cameras, just say goodbye, say thank you.
Okay, thank you so much for this, all these wonderful questions.
Thank you. Bye-bye.
Bye-bye.
Thank you.
Bye-bye.
Bye.
Bye.